G-protein coupled receptors, also called metabotropic receptors, are membrane-bound proteins that activate G proteins after binding neurotransmitters. Like ionotropic receptors, metabotropic receptors are primarily located along the dendrites or cell body, but they can be present anywhere along the neuron if there is a synapse. Metabotropic receptors are also important for receiving incoming information from other neurons. G-protein coupled receptors have slower effects than ionotropic receptors, but they can have long-lasting effects, unlike the brief action of a postsynaptic potential. G-proteins are enzymes with 3 subunits, alpha, beta, and gamma. In the resting state of the G-protein complex, the alpha subunit is bound to a GDP molecule. There are multiple types of alpha subunits, and each initiate different cellular cascades in the neuron. When a neurotransmitter binds to a G-protein coupled receptor, the receptor is able to interact with an inactivated G-protein complex. The complex that binds is specific to the receptor. Different metabotropic receptors for the same neurotransmitter can have different effects in the cell due to which G-protein binds. Once coupled to the receptor, the GDP molecule is exchanged for a GTP molecule, and the G-protein becomes activated. After activation, the G-protein complex will separate into the alpha GTP subunit and the beta-gamma subunit. Both components can alter the function of effector proteins in the cell. Effector protein functions can range from altering ion permeability across the membrane by opening ion channels to initiating second messenger cascades. Second messenger cascades can have long-term, widespread, and diverse cellular effects, including activation of cellular enzymes or altering gene transcription. In certain situations, the activated beta-gamma subunit can open or close ion channels and change membrane permeability. Muscarinic acetylcholine receptors in the heart use this pathway. When acetylcholine binds to a muscarinic receptor in the heart muscle fiber, the activated beta-gamma subunit opens a type of potassium channel called a G-protein-coupled inwardly rectifying potassium channel, or GERK channel, hyperpolarizing the cell. This inhibitory effect explains why acetylcholine, or an agonist like atropine, slows the heart rate. In addition to direct effects like the activated beta-gamma subunit opening ion channels, G-proteins can have many indirect actions in the cell through the use of second messenger cascades. The specific second messenger pathway that is activated or suppressed by G-protein action depends on the type of alpha subunit. For example, norepinephrine can act on either alpha or beta adrenergic receptors. Beta adrenergic G-protein coupled receptors couple to a stimulatory G-protein, or GS, which initiates the cyclic AMP second messenger system by activating the enzyme adenylyl cyclase. Alpha-2 adrenergic receptors, however, couple to an inhibitory G-protein, or GI, and suppress the activity of adenylyl cyclase. Alpha-1 adrenergic receptors couple to a third type of G-protein, GQ, which activates the Phospholipase C pathway. One neurotransmitter can, therefore, cause a wide range of cellular effects after binding to G-protein coupled receptors, unlike the single function of ion flow through the ionotropic receptors. The pathway initiated by norepinephrine will depend on the type of receptor a specific cell expresses. The cyclic AMP second messenger pathway is used by many G-protein coupled receptors. Activation of the pathway is caused by the GS alpha subunit and inhibition of the pathway is caused by the GI alpha subunit. When activated, adenylyl cyclase converts ATP to cyclic AMP in the cytoplasm. Cyclic AMP then activates another enzyme called protein kinase A, or PKA, by binding to the regulatory subunits, allowing the catalytic functional subunits to separate and become active. Protein kinases add a phosphate molecule to proteins, a mechanism called phosphorylation. The addition of the phosphate changes the activity of the protein and how it functions in the cell. The end effects of this pathway will depend on which proteins are targeted. For example, cyclic AMP can gate ion channels, and PKA can phosphorylate ion channels, altering permeability and membrane potential. Phosphorylation can open the channel, or it may modulate the activity of the channel, making the channel easier to open or remain open longer. In addition to altering ion channel function, PKA can phosphorylate other proteins important for neuron function, such as proteins involved with neurotransmitter synthesis and release. One other critical target of PKA phosphorylation is the transcription factor CREB. Transcription factors bind to DNA in the nucleus and change the rate of gene transcription. Phosphorylation by PKA can cause CREB to initiate transcription of genes, creating new proteins for the neuron. Depending on which genes are transcribed, the effects on the neuron can be long-lasting. Overall, neurotransmitters working through G-protein-coupled receptors and second messenger cascades like the adenylyl cyclase pathway can cause a diverse range of cellular effects, from opening ion channels to changing protein activity via phosphorylation to altering the protein synthesized in the neuron. The GQ alpha subunit initiates a separate signaling pathway in the cell by activating phospholipase C. Phospholipase C targets PIP2, which is a phospholipid present in the plasma membrane of the cell. PIP2 is split into two cellular molecules, IP3 and DAG. DAG remains in the membrane and interacts with protein kinase C, PKC. IP3 moves to the endoplasmic reticulum, where it opens calcium channels and allows calcium to flow into the cytosol. Calcium is also a second messenger in the cell. One important effect is the binding of calcium to calmodulin. This complex can then activate another kinase, the calcium calmodulin dependent protein kinase, or CAMK. Both PKC and CAMK can phosphorylate specific cellular and nuclear proteins like PKA. One characteristic of G-protein coupled receptor activation is the signal amplification that takes place. One receptor is able to activate more than one G-protein complex. The effector proteins activated by the G-protein can create many second messengers, and the activated protein kinases can each phosphorylate multiple cellular proteins. This means that one neurotransmitter can have a significant effect on cellular function. Eventually, the cascade initiated by binding of the neurotransmitter to the G-protein coupled receptor needs to end. The alpha subunit of the G-protein is able to convert the bound GTP back to GDP after a short period of time, inactivating the G-protein. The alpha subunit will then interact with a beta-gamma subunit and stay in the resting state until activated by another G-protein coupled receptor. Enzymes in the cell, called protein phosphatases, find and remove the phosphate groups added to cellular proteins by the protein kinases. And finally, other cellular mechanisms exist to remove calcium from the cytoplasm and degrade other second messengers.